Good evening, everyone. I'm Carmi Garzione, Dean of the College of Science, and I just want to thank you all for joining us at the opening night of the 2026 lecture series. Before we begin tonight, please join me in giving tonight's musician, Carlos Arzate, another big round of applause. Carlos. Well, to set the stage for this year's lecture series theme, Today's Science, Tomorrow's World, Building a Better Future, I'd like to ask the audience to take a moment with me. Please take a moment to imagine a world where neurodegenerative diseases can be stopped or even reversed. Imagine a world where people live well beyond 100 years old and are healthy and active throughout their lifespan. Imagine a world where energy is abundant, low cost, and clean. Imagine a world where humans constantly leverage artificial intelligence to solve myriad problems that could never be solved before. These are the imaginations of scientists. I suspect that virtually everyone in this room imagines these types of futures. If only we could see what the world would look like in hundreds, thousands, or even millions of years into the future. Scientists often do their work knowing that they are unlikely to see the future that their work will lead to. I believe that every person in this audience and in the world has an inner scientist. We are all curious about what is possible and how we could achieve it. This year's lecture series aims to share that curiosity, that imagination of a better future that is the literal engine of scientific research. It's great to see so many people in the audience as we celebrate our 20 years of the College of Science lecture series. It was right here in Centennial Hall in the spring of 2006 when the series began on the topic of evolution, bringing together educators and researchers from across campus at the University of Arizona. The lecture series has flourished for 2 decades with tens of thousands of attendees over the years. Now more than ever, we are deeply grateful to have your support of the sciences and this lecture series. And I'd like to just take a moment to recognize the Galileo Circle members in the audience tonight. The Galileo Circle is a community of individuals whose support is vital to the continued excellence of the College of Science. Thank you for your continued support of our students, faculty, and staff, and thank you for joining us here tonight. I'd also like to thank Arizona Arts Live for their continued partnership with the lecture series and for helping us to bring these talks to life each year in Centennial Hall. This really is an art form, figuring out how to communicate science. So we really appreciate this collaboration with the arts. I'd also like to thank our corporate partners for the lecture series, in particular our presenting series sponsors, the Marshall Foundation and Research Corporation for Science Advancement, and our series sponsors, Holualoa Companies, O'Reilly Chevrolet, South 32, Tech Launch Arizona, Tucson Electric Power, Tucson Medical Center, and Visit Tucson. We are deeply grateful for your support. And I'm pleased to share that the College of Science is collaborating with the University of Arizona Poetry Center once again for this year's lecture series. In our spirit of trying to reach audiences through many different platforms of communication, a poet will read an original poem crafted for our lecture. We hope that this reaches some of our audience members in ways that a science lecture could not possibly do that. So tonight's poet is Peggy Shoemaker, and Peggy is a Tucson native who served as the Alaska State Writer Laureate and received a poetry fellowship from the National Endowment for the Arts. Peggy is a professor emerita of the University of Alaska Fairbanks and the author of 8 books of poetry, including the most recent entry, Stillwater Carving Light. We are delighted to have her with us here tonight. Please join me in welcoming Peggy to the stage.
Before I read a poem for you, I would just like to voice my gratitude for scientists. Thank you for the intricate and imaginative attention that you pay. Forgive. They forgive us a lot, these bodies. They forgive, but they don't forget. How well do we forgive them? Our cells that can't control themselves, our bewildered nerves that in their torment us. Forgive us our excesses as we forgive diseases we did nothing to deserve. Deliver us from quackery. Give us this day deep gratitude. Let us respect those who pay close attention, those who know each mystery revealed reveals deeper mystery. They forgive us a lot, these bodies. They forgive, but they don't forget. Thank you.
Well, thank you, Peggy, for that thought-provoking poem. That really is a nice way to think about the topic we're going to discuss tonight. And so I'd now like to introduce tonight's feature speaker, Dr. George Sutphin. George is a faculty member in the Department of Molecular and Cellular Biology at the University of Arizona, where his research focuses on genetics of aging. He earned his bachelor's and master's degree in aeronautics and astronautics from the University of Washington. Talk about a career shift. Followed by a PhD in molecular and cellular biology. And so following the completion of his PhD, George conducted postdoctoral research at the Jackson Laboratory, where he further advanced his work on molecular and genetic mechanisms that influence aging. He joined the University of Arizona as faculty in 2018 and has continued to build a research program centered on understanding the biological processes that drive aging and age-related change in our bodies. In addition to his academic appointments, George serves as the CEO and chair of the board of directors of the American Aging Association and was recently re-elected to a second term. His contributions to the field have been recognized through election as a fellow of both the American Aging Association and the Gerontological Society of America, as well as with the Galileo Circle Curie Award from the College of Science and the University of Arizona Early Career Innovation and Entrepreneurship Award. Without further ado, let's welcome George to the stage.
Good evening, everyone. I'd like to start by saying thank you to those of us who are in the trenches every day doing science. It's really encouraging to have all of you out here showing your curiosity and your enthusiasm. So thank you for being here and supporting science at the University of Arizona, please. Before we get into today's topic, I want to say, since we're at the opening night, a little bit about the series as a whole. I'm really excited about the series. I think you'll see a lot of overlap and a lot of shared themes We're all very curious. We're very optimistic about the future. I think scientists tend to be that way naturally. And I think you'll see these themes come through. And I really hope that our passion and enthusiasm for the work that we're doing and the topics that we're going to share with you comes through, and that you can share in that with us. So today we're going to talk about aging biology and how, by targeting aging biology with medicines and other interventions, maybe we can push back the late life disease and frailty and have longer, healthier lives. Next week, we're going to learn how fruit flies are teaching us things about how our brains work and maybe can be used to develop new treatments for neurodegenerative disease and other brain disorders. You'll see a lot of overlap between this talk and the talk today. My lab studies a roundworm called C. elegans, and we are using that worm just like they're using the flies to learn about the biology that is relevant to humans. So we'll get into that. Then we're going to talk about fusion energy in the current state and how we might be pushing toward a really clean, renewable energy source that could change energy generation and support many of the activities we do as humans. And finally, we'll learn about planetary science, so really zoom out to the whole planet, but also how artificial intelligence fits in with our daily lives, but also the science that we do. It is more and more a center of our world. So I like this set of topics in particular because I think that all of these share a common theme. Up until fairly recently in our scientific history, all of these have been really driven by basic science. And here, this lecture series is really about basic science and the value of basic science and how that drives innovation and the things that we use in our daily lives. But more recently, these are all getting to the point, these fields, where we're starting to just see the edge of the benefit from studying these things. So we're getting closer to fusion power, we're getting closer to seeing the end result. And so these topics really illustrate, the topic of the talk, the things that, how basic science is going to drive the next things that change our world. Now, a word on basic science, since that's the theme, and that is the major theme of the lecture. When we think about scientific endeavor, oftentimes what we think about is the things that come at the end. So the medicines that are going to extend our lifespan or treat neurodegenerative disease. Maybe the fusion power plant that's going to power our world in the future. Or the ways we can learn to better take care of our environment and our planet, and maybe the next chatbot or the AI that's going to help drive these. But these are really what I consider the tip of the pyramid. These are the products, the end result, the goal. But to get there, there is a long process. If we start at the other end, first we have to have an idea. Maybe we don't even know what that end product is going to be in the end. Maybe we do. But maybe we're just curious. We see something out in the world and we think, that's interesting. Let's go see what that's about. So then we start to investigate it. We start to learn about it. And then we develop that idea into some starting idea of how does the world work? Why does it work that way? Eventually we get to the point where we start to understand it enough, where we can refine our ideas to the point where now we're moving almost into more engineering. How do we start using this to do useful work in the world? And then that brings us all the way back around to this consumer product that we're trying to do that makes the world a better place. But it's really these first two layers where basic science, we need a lot of work. Not everything we do at the idea stage is going to become a product. There's lots of things that are dead ends. But so we have to do a lot of this initial work, the idea development, before we can get to these useful products. And that's really what we're about in the College of Science. Ultimately, we hope to get to the products. Today, we're going to talk about developing medicines to treat aging and age-associated disease. And this process has a little bit different workflow. This is more specific. So we have a discovery phase where we're trying to understand the biology that drives aging. We figure out how it works. Ultimately, we come up with a way that we think we can intervene in that, and then we do preclinical testing, often in a species like mouse, where we see if we can make that mouse live longer, healthier, more functional in the later years. And then ultimately, we get to the point where we can test that medicine in a human and do clinical trials. Aging medicine is fairly new. We are really, most of the field is either in the discovery mechanism or preclinical stage. I'll talk about this later, but we are just now at the point where the first human trial for an anti-aging drug is now underway. So we are really at the point where we're just starting to see the tip of that pyramid. So, how do we experience aging? Aging is truly the universal thing in health. We all experience it. We may not experience it the same way, but we all go through the process. There are certainly good sides to aging. As we get older, we develop longer relationships, bigger families, we make friends that we have for years or decades. The meaning in our lives deepens. More independence, so we learn new skills, we can do more things. Oftentimes it comes with additional financial independence, so we're able to go spend time with the people we want to spend time with and do more of the things we want to do. And wisdom. We maybe become more comfortable in the world, maybe more comfortable with ourselves, and more willing to put some of the more unusual parts of ourselves out there. So all of these things are wonderful. These are the good parts about aging. But of course, that is not all there is. There is a darker side. On the more mundane, we start to experience more daily friction, maybe aches and pains, or our eyesight starts to fail. Just everyday life gets a little harder because parts of our bodies are not functioning as well as they used to. As this progresses, maybe we lose some of that independence, and it's undermined by not being able to walk as well, or needing help crossing the street or getting out of a car, that sort of thing. And ultimately, underlying all of this is the aging processes that's driving frailty and disease. So, my interest is how do we get more of those good things and less of the bad ones, right? That is the goal. And I am a scientist. You heard about my professional life, but I am also a human, and I am aging, and I have personal motivations. And I always like to share just a little bit about that. I run a lab at the University of Arizona. We have students at all stages, from undergraduate up to graduate, professional staff, and so on. And we really focus on building the community. My philosophy is if you want to work with the people you enjoy hanging out with, right? So we have a great group. We've got about 20 people there now. We've had many, many people come through the lab over the last 10 years. Outside of the lab, I have a family. My wife, Ashley, son, Callum, they're in the audience today. They're able to join us. And my mom, who just retired and moved to Tucson 2 weeks ago, is now able to be here tonight, so she's here. Thank you. I have many long friendships. We like to do hiking, play board games, play video games. A couple of my good friends are here tonight as well. And I'm a dog person, so we've got, this is Doppler and Zelda. They are the other place I like to spend time. So why am I telling you this? This is my motivation. These are the people and the activities and the family that I want to have more time, more functional time to spend with. And that is the real motivation behind why we study aging science. So let's get into it. I'm going to put up some of the most common diseases in our society. So heart disease is currently number one. How many of you out in the audience, just raise your hand, know somebody or have been affected by heart disease in some way? You can see that's most of the audience. How about cancer? Right. Neurodegenerative disease. How about kidney disease? And how many of you know someone who individually has been affected by more than one of these at the same time? That's quite a bit. Comorbidity, getting multiple diseases at the same time, that's very common with aging. So I put these up here because these, heart disease and cancer, number 1 and 2. Alzheimer's disease is predicted to be the number 1 cause of death within the next 20 or 30 years. So these are all the major causes of death in our society. And what they share is this one feature. So what I'm showing you is a graph where the vertical axis is your risk of getting that disease. The other axis is how old you are. And what these diseases all share is a curve that looks like this. So as we get older, we are more likely to get that disease. And we're not just a little bit more likely, this is an exponential curve so that every year you're more and more and more likely to get that disease. And it's not just heart disease. All of these major causes of death in our society share this. It's something like 7 out of 10 of the top causes of death. It accounts for somewhere, depending on how you calculate it, between 60 and 90% of mortality share this feature. How old you are is the major risk factor for getting the disease. And it's not small. If you compare age as the risk factor to any other environmental thing, exposure to a chemical or diet or alcohol consumption, age beats them all hands down by a big margin. It is far and away the biggest. So what that tells us is if age is the major risk factor, our biology changes as we age. And what my thesis is, and the reason that I study aging, is if we can understand that biology and do something to intervene in it, we can now potentially have an outsized effect on many, many of these common diseases and push back our healthy lifespan into the future. Let's run a thought experiment to demonstrate the potential for how big of an effect we might have. So here's the thought experiment. Imagine someone, a woman today who's 50 years old. So the life expectancy is 81, so she currently has 30 additional years on average to live. Now, if we were to completely cure cancer today, 100%, nobody gets cancer ever again. My question is, how many additional years of life would you expect this person to get? If you think it's between 0 and 5, raise your hand. Got some in the 0 to 5, got 5 to 10. How about 10 to 15? 15 plus. Maybe out there. It's about 3 years. Heart disease is about 3 years. Now, why is this? It's because if you cure cancer, you are still at an age where you're at high risk for heart disease, or kidney disease, or Alzheimer's disease. And so curing one of these diseases in total, even one of the top 2 killers, will only give you a little bit of extra time. If we do both of those, now you're doing a little better. Maybe you can get 5 or 6 years. Add in diabetes and stroke, now you're up to maybe 10 or 15 years. But if we were to, right now, be able to affect human aging the same way we can do in laboratory animals like mice or roundworms or fruit flies, this is the effect that we can have. 20 or 30 years, that range, it's a big effect. So I want to show you the data behind this and to see what it looks like. And to kind of prove the point. To do that, we're going to look at a type of data called the survival curve. I look at these all the time, but I know if you're unfamiliar with them, sometimes it helps to just to see what the process is. So what these work, on the vertical axis, we have percentage alive. So this is a population level metric, so what percentage of that population is alive? And on the other axis is age, so at each age. So as you might expect, the farther out into older and older age we get, the fewer and fewer people, the lower percentage of that population is alive. So if we were to do something to shorten lifespan, what you'd see is a shift to the left. And if you were to lengthen lifespan, you see a shift to the right. We are, of course, more interested in the increase in lifespan. Shortening lifespan can be informative, but there are lots of things you can do to influence lifespan in the shortening direction that have nothing to do with aging, exposure to a chemical toxin, for example, or an accident. Whereas if you increase lifespan, you almost by definition have to have done something to slow the aging process. And so that is what we focus on primarily. And this idea of lifespan, this is the major type of data that we look at as our kind of first pass. We look at a lot of other things, but this is the first pass one that we look at first. So why do we think that aging and targeting aging can really give you this big benefit over curing a single disease? The big one is, or one good example, and this was the first thing we found that really extended lifespan, and that is this idea of dietary restriction. This is simply reducing total food intake without being malnutritious and remaining adequate nutrition. This might look like in a mouse, you can just reduce the number of food pellets you give them by 30 to 60%. It also includes things like fasting. It includes things like sugar restriction. It's a pretty broad term. The data I'm going to show you is just total food restriction. So here's what it looks like in a mouse. So this is the survival curve I just showed you. Now it's a mouse one. The blue and the green are fully fed mice. They can have as much as they want. The one on the right, I believe this particular graph shows about a 40% caloric restriction. So you look at what the control mouse ate, and you take away 40% of that food, and that's what you get. This gives you a huge increase in lifespan. This one is probably 40 to 60%. That's half again as long as we currently live if we're a human. This data is why, and we think this is targeting aging for a variety of reasons. And this is why we think if we can do the same thing in the human, we can have a much outsized effect to even curing a big disease. So that is the scientific basis for this idea. What's nice is this works in everything we try it in. So here there we go. Everything from a single cell yeast, fruit flies, the roundworm that I study on the lower left, and mice. And it works in probably 20 or 30 other species that have been tested. What's nice is we want to make sure that we're not just extending lifespan without making the animals healthy, right? So we don't want to extend that very end of life where you're suffering and you have disease. What we want to do is extend the healthy, functional part of life. And it looks like most of the things we do that extend lifespan do that. So here is just looking at rats, and this works in monkeys. These are rhesus macaques. In both cases, you can just see visually that the animals on the right who are dietary restricted at the same age they have healthier coats, they have better posture. The monkey looks a little angry. He probably knows he got the short end of the stick on this deal, but in any case, he's healthier if he's angry. That actually gets to the point that this may not be the best intervention for humans. Humans like to eat, and convincing the population as a whole to reduce their caloric intent by 30% may be a challenge. There are people who do it voluntarily, though. But this is the thing that kind of works the best so far. So I hope that I've given you an argument for why we study aging. Let's take this from a different perspective and maybe look at our history of humans and how long we've lived in the past, and then what is the potential for aging to affect the way we currently practice medicine. So this is going to be more lifespan curves. We're back to it. Let's look back at our prehistory. And I'll also call this pre-medicine, so 50,000 years ago. This is more what our lifespan curve we think looked like, where most people die when they are young. And the reason for this, we still got we think people still lived out into their 60s, 70s. It was just rare. And the reason is that we had exposure to predators, more infection. We were out in the wild, so there's more weather. So we have all these extrinsic factors, these outside factors that are causing most of the risk to our lives. As we get into agriculture and start to live in cities and build civilizations, we start to do better. You still have a lot of early life. A lot of this is driven by infant mortality, which we really didn't figure out and start to do much better at until just recently. But even here, most of the population was still kind of living into their 30s and 40s, maybe. And so we're maybe less exposed to some of these external factors like predators and weather, but we are still not perfect at it yet. I would call this early medicine, where maybe you have a healer in your village. A lot of the knowledge is trial and error. It's passed down from tradition, but it's maybe not systematic, and it's kind of hit or miss. Then we do a big jump all the way up to the last 100 years. We had a revolution in medicine driven by things like the germ theory of disease, where we realized that things like bacteria and viruses cause disease, that they can spread, and we can stop that spread through things like hygiene and treat it with things like antibiotics. And this really had this effect, which is it pushes our lifespan curve out toward the older ages, and it also has this effect we call squaring of the curve, where most people now tend to live later in life, and then it's the later life diseases that are really causing the problems. And so now, instead of external factors, we've gotten pretty good at even dealing with infection as a whole. I know we just went through a pandemic, but infection, we are not exposed to the weather so much. We don't run into predators all that often. And so most of what harms us today is problems with our internal biology that happen as we get older. And this is really driven by aging. This is the diseases that we have talked about: heart disease, cancer, neurodegenerative disease. The shared bit about all of these medicines up until this point are they're reactive. So what happens when you get sick now? You wait until something goes wrong, you go see your doctor, they try to fix it. That is mostly how we do medicine and how we've done it in the past. But the promise of aging medicine is that it remains scientific, so that was the driving factor. We've had a systematic scientific way to understand how biology works and treat medicine, so that remains. But now we move from a reactive system to more of a preventative system, where we understand what's going to happen to our biology, we can detect it earlier, we can start to treat it, and then have an outsized effect, push back these diseases, maybe prevent them. That is the promise of aging, and what I think, once we really get going, could lead to a revolution in medicine. We see hints of this already in the preventative medicine movements. Okay, that is why we study aging. That's why I study aging. Now I want to talk about both where we are in our understanding of aging biology and why basic science is particularly important now and particularly in the near future. So what is the molecular biology that drives aging? A few years back, this field, like what I would call modern aging medicine, really got started in like the late 90s, or late 80s and early 90s, when we really started to be able to do genetics really well, and this became kind of a focus, the molecular biology of aging. And since that time, we have learned a lot. So we know now about what we now define as hallmarks of aging. So these are molecular processes in our cells that share two features. A, they degrade with age, so they stop functioning as we get older, and that's part of it. But in addition to that, that change with age causes disease. So they have to be causally in the aging pipeline. They have to be causing aging, not just like changing with age. Right now, there are 12 of these. In 2013, when the first came out, I think there were 9, and now there are 12. So even in the last 10 years, we've added to this. That might show you the rate of change in the field and how early we are. And here you might see some things that you recognize. So here, mitochondrial dysfunction. Mitochondria are the powerhouses of our cells, so our ability to produce energy. We already talked about dietary restriction, so nutrient sensing, having problems with that is a problem. A genomic instability. Our cell's ability to maintain our genetics and keep the sequence intact and not turn into things like cancer. I want to go through, I would love to go through all of these, but we don't have time. But I'm going to focus on one just as an example. How many of you out here have heard of telomeres before? Quite a few hands. This is probably the one that is the most public-facing. Most people have heard about it to some degree. And this is certainly a cause of aging. So let's look at that in more detail as an example. So what are telomeres? Our DNA, our genes, are organized into chromosomes. And each of these chromosomes has an end cap on them, which is these telomeres. So telomeres are basically a repeated sequence of gene. They repeat thousands of times right at the end. And they act as sort of a protective cap on our chromosomes. Oftentimes, the analogy is made to shoelaces. A little cap on the end of the shoelace keeps it from fraying, makes it work better. The thing is, every time our cells divide, it does not extend all the way to the end. It loses a little bit of that telomere. And so as our cells divide more and more and more as we grow and age, eventually it gets to a point where the telomere is too short and it can no longer divide. So this is, and this is kind of the early model of what these things were. So that's great, right? So maybe what aging is, and this was one idea when this first came out, is that we lose the ability for our cells to divide, and maybe if we can just lengthen those telomeres out, we can recover and become youthful again. So you think this will work? Good. I might have given the punchline away, yeah. It does work in almost every many of the science fiction novels you might read. This is the most common way to do this. I'm a science fiction fan, so I have to put that out. And if you're gonna do a science fiction and you want someone to live longer, come talk to me. It's not just telomeres. So let's take a look. So maybe the next way we can refine our model, so our initial model maybe is that more telomeres is longer life. Okay, but that lets our cells divide. What do we think about when we think of a disease that's associated with cells dividing too much? That's cancer, right? Yeah. So maybe our next model, and maybe this gives you some insight into how scientists think, and we change our thinking about these processes and evolve them over time. So now, maybe telomeres, the reason they exist is they're a trade-off between letting our cells do important processes like heal wounds and grow and maintain their health, things that you need cell division for, and cancer, which is out of control cell division. So maybe they're a balancing act. Okay, so now we have an idea that we can test. What if we tip that balance and we do longer telomeres? We can do this. There's a gene, an enzyme called telomerase, which, if you express it in a cell, it gives that cell the ability to make longer telomeres. And so we can give that a mouse, that cell, that ability, and see what happens. Okay, so let's look at maybe a lifespan prediction. Let's say we give longer lifespan. Maybe our prediction here is that we are going to get longer-lived mice. Does that work? Turns out that works. So you express on the blue and the gray, those are mice with normal-length telomeres. And then the mice in the black line, these have this enzyme that lets them make longer telomeres. And it works. It's not going to cure aging. They are still aging, but they get a little bump in lifespan. So probably this tells us that it's important for the aging process. Now we look the other way. What if we shorten telomeres? Well, then our prediction goes the other way. Maybe we shorten lifespan. That works, but there's a caveat. So the red line is mice with shorter telomeres. The green line is mice with normal telomeres. But it turns out that the intervening mice also have short telomeres. And it takes not the first generation, so the mice offspring and their grand-offspring and their great-grand-offspring and their great-great-grand-offspring. It's their great-great-great, I think I have that right, grand-offspring that actually start to show a lifespan effect. So this was kind of a surprise that if you get rid of the ability to make telomeres, it takes 6 generations before you start to see an effect. So I'm going to mark this one as, yeah, it worked, sort of. So we're getting closer. So maybe the model is right to some degree, but maybe not right. But now we can also make predictions about cancer. More cancer for longer, because the cancer cells will also have this enzyme, the ability to make cancer, and less cancer for shorter ones. So this is just a bar graph, so the higher that bar is, the higher percentage of that group that has cancer. Turns out there was no effect in the lengthened telomere group, so that one I kind of marked as neutral. It wasn't like true or not true, just there's no effect. And it turns out on the other side, if we look at the shortened telomeres, they didn't have less cancer, they had more cancer. So what's the point of telling you this story? I think the main thing is that it looks like telomeres are important for aging. That's why they're on the Hallmark of Aging list. But I wanted to give you a sense for how, as scientists, we come up with an idea, a model that we think, okay, maybe there's this cancer versus growth idea, and we're going to make predictions, we're going to test those predictions, and then we're going to make our model better based on the answers. That's what we do here. So now, telomeres, this is still fairly old news. Telomeres we've been studying for years. There's still people studying telomeres. We still don't understand them fully. But they are important for aging, and we are still in the process of learning that basic biology that drives them. That is true of all of these hallmarks. We could do a similar exercise through all of these. We could see what our early predictions, which ones of those bear fruit and which ones are maybe not quite right. We can then make a better model, and over time, we're going to understand these better. So we have these 12 areas of biology where we think, we know that these are the ones that we are very confident that they are involved in aging. There's probably more out there. But I want to now ask, how well do we really understand the biology? So I've given you a setup for the telomeres, like we aren't quite there yet on understanding them. Okay, let's find out. Maybe let's go to the news and see what the news says about this, all right? Reverse aging in dogs, did it in mice, we're almost to humans. So my work is done, I think, so looks like we're done. I'm being a little hyperbolic, of course, but in reality, these headlines come out all the time. There are more realistic headlines, but there's no shortage of the ones. And if anyone says they've reversed aging, pretty much in any model, the answer currently is no, at least I'm not aware of it. And we've certainly not done it in humans. So how well do we really understand aging? I'm going to use an analogy. And we're going to compare aging biology today to geography. And this will make sense why I'm using geography. This is an analogy by my graduate mentor, actually, Matt Kaberlein, who's currently at Optispan in Seattle. And he came up with this, and I think it's really illustrative. So, geography today. We have Google Maps, right? We know the shape of our world, we know what all the coastlines look like, we have some idea of weather, we can zoom in, we can get directions from the airport to Centennial Hall, and we know every turn along the way. We even know that we need to turn left instead of right because there was an accident 10 minutes ago. This is the level of detail we want in aging biology, in all of these areas of science, right? That level of granularity. But that's not where we're at right now. Maybe that's not a surprise. The analogy is that we're closer to the map of the world in 500 BC. So, Hecatius's world map. So, I'm not saying this is useless. We know the shape of the coasts. We know where the rivers are. We know where the mountains are. We can use this to navigate from city to city, maybe from village to village a little, get to countries. But on the other hand, there's missing continents. Those might be analogous to the hallmarks of aging. And we certainly don't have the level of detail we need to go in and make targeted, precise changes, which we would want to do in the context of something like aging if we want to improve our health and be safe about it, right? So this is really closer to where we are. I mentioned that the field is relatively new. 1980s to like 1990s was really when the current modern molecular biology of aging got started. And we've come a long way, but there's a lot of distance to go yet. And that kind of brings me to my work and what we're doing at the University of Arizona. So I'm in the College of Science, Molecular and Cellular Biology, and we're really interested in understanding these basic processes. So what I do and what I'm interested in is I'm going to mark these. This is what we know, right? I am interested in understanding what we don't know, finding those other continents. So how do we do that? The problem with studying aging as humans is we live a long time. So human lifespan is about 80 years right now on average. Human studies are expensive, and you really don't want to just go in and do experimental biology directly on a human unless you have a pretty good idea if something's going to work, right? So what do we want? How do we fix this? So what we do start with human data. We know a lot about human biology, but we don't necessarily know which of this biology affects aging. So now we can generate some candidates. So which genes do we think might have an effect on aging? Maybe we know they have an effect on diabetes or cancer or these other things. Maybe we see another process. What drugs might be interesting to test for aging? What environmental factors? So I consider dietary restriction more of an environmental factor, for example. Then we take those candidates and we go to a model where we can do studies quickly and inexpensively. And the model we study is called C. elegans. It's a roundworm. They're about 1 millimeter long, so they're small. They're very easy to grow in the lab. And now what we can do is we can test in that worm which of these interventions extend lifespan and affect aging. Once we have some hits, the ones that work, we can then figure out what they do. We have lots of tools for genetics and molecular biology. Once we have an idea of what they do, and if that idea is something that we think would also work in a mammal, then we can take that into a system like a mouse and validate that idea. Does it work in the mouse? Does it affect the same molecular processes? And ultimately, we want to bring that back to humans, develop medicines that have data and evidence for safety and effectiveness in mice, and then take those into the human and see if we can actually affect the diseases and the function with age. So, why worms? Why not start in the mouse? There's really a couple of reasons here. One thing is mice are one of the shortest lived mammals. They're a lot closer to our biology, but they still live 2 to 3 years. So if I started an experiment today, we're probably not going to have an answer to that until maybe 2029 or 2030, if I wanted to test a drug, for example. Worms live about 3 to 4 weeks. So I will know by March. So that's a 40x benefit in terms of speed. The other side is cost. Yes, and this is real. So if you want to do a full lifespan study in enough mice to get reasonable answers, and you want to learn something about their health, it costs somewhere on the order of $200,000. You have to keep those mice in a controlled environment, free from pathogens, very standardized food and care. It's expensive. A worm, we can do that in about 20 minutes, or $20. So that's about a 10,000-fold advantage. And then you might ask, but okay, does aging in a worm mean the same thing as it means in a human? That's a really good question, and at some level, it's not going to translate perfectly. Worms don't have like a heart or a liver or a kidney, for example. They use other cellular systems for these processes. But if you look in the subcellular level and you look at the molecular processes that drive aging, that's the hallmarks of aging. These are shared to a large extent between worms and mice and fruit flies and yeast. And so many of the things we discover in the worm, and look at these numbers. If we only 10% of those translate, we're still way ahead of the game. And in reality, it's higher than that. So that is why we study the worm. So how do we do it? The old way is these worms live on a little Petri plate. You sit at a microscope. They look something like this. And yes, if you've done this for 8 hours at a time, you will have nightmares about it. Personal experience. And you sit there with a little piece of platinum wire, and you go in and you bonk every worm on the head with that wire, and you ask, is that worm alive? If they move, they're alive. If they don't move, they're dead. It's not rocket science here. I was a rocket scientist before this. So this is tedious. Maybe an experiment has 1,000 worms, and you need to look at them every day until they die. So this takes a lot of effort. Now what we've done is we've tried to automate this. I have an engineering background, so we do engineering in the lab. We've basically built a robotic platform. This is if you're familiar with computer-aided wood design, this is a machine that does 3D computer-aided wood cutting. We just take the cutting tool off, we put a microscope on there. So a camera, light source, and lenses. We stick that in an incubator to control the environment. And then instead of using that platinum wire to poke the worm in the head, we now shine blue light on them, and they don't like that, so they move, and then we get our motion data. And we can do about 20,000 worms at the same time. And we have 3 of them right now. So this is what it looks like. Up on the left, you have young worms. On the right, you have old worms. And what you'll see, that flash was that blue light going off. And what you see, I'll let it run one more time. But what you see is the worms are kind of moving around, not the old ones so much. They do have less activity. They are aging. And then when they see that light, now we get a big movement. Our computer software can now analyze that, figure out when the worms stop moving, and give you a lifespan. And you get activity data. So you get a bonus of how active that worm was during the lifespan. And it runs this every day. And we've probably run 300,000 to 500,000 worms through this thing so far. So this lets us do high throughput screening. The biggest study we've done so far is maybe on the order of hundreds of genes or hundreds of drugs. But if you scale this, you can imagine doing up to millions. And there are millions of compounds out there that we could test as potential drugs. We feed a population of worms each of these drugs. We look for the ones that extend lifespan. And then these give us our targets where we can spend that money that we want to on the mammalian system and see if we can extend lifespan. So let me give you one example. We did a genetic screen so we can knock down genes. We started with human data, and we found this pathway. So what does this pathway do? The blue at the top is tryptophan. You may have heard of this famously for the thing in Turkey that makes you sleepy on Thanksgiving. Turns out that's a myth. Turkey isn't particularly high in tryptophan. Tryptophan is used to make melatonin, which makes us sleepy in our bodies. But my theory is it's the other 10,000 calories you eat at Thanksgiving that makes you sleepy. That might just be me. Okay, so what did we find? So it gets turned into a lot of things, but one of the things it gets turned into is this molecule called 3-hydroxyanthranilic acid. It's a mouthful, or 3-HAA. That's the orange one. There's two genes. The gene kinu makes 3-HAA. The gene hao consumes 3-HAA, degrades it. And what we found is if you knock out the hao gene, which you knock down the thing that is degrading 3-HAA, so now it's no longer degraded, it builds up, you get a big lifespan extension. So the red and the blue are the lifespan for the knockout worms. So these are lacking this gene. They build up 3-HAA. You can also just feed 3-HAA to the worm. That's the yellow curve, and they are also long-lived. The worms are transparent, which is like a handy thing, so we can actually see what's going on. And they actually turn red. The 3-HAA is red in color, and there's so much of it building up in the worm that they change from kind of a translucent to a red color, right along the intestine. So this seems to be a buildup in the intestine. We've done a lot of work to figure out what it's doing. I'm not going to go through all of that. I'm going to give you one little taste of some of the tools we have to learn about these things. So there's these proteins from jellyfish. So they're a gene. Genes use a universal language of DNA, and so we can take a gene from the jellyfish and put it in the worm, and we've attached it to these two genes, the thing that makes 3-HAA and the thing that breaks it down. One's in green, one's in red. And then you can take pictures of the worm. They're transparent. And this is beautiful. I mean, that's what one of these worms looks like. If you know about worm physiology, you'll know that that green is hypodermal tissue and the orange is neurons. So it looks like these are happening in different places, hao in neurons, kinu in GFP. So what do we think is happening with these genes? So it starts in the hypodermis. Tryptophan gets turned by kinu into 3-HAA. We think this then travels to the intestine, so our stomach tissue, the worm's stomach tissue, and then ultimately some of that travels to the neuron where it gets broken down by hao into another molecule. But what we think, a lot of the action for this molecule is happening in our intestine, and we know it does a few things. So it just generally improves the health of those cells, so the cells are more robust. It also helps those cells fight bacterial pathogens. It also makes those cells resistant to other forms of stress. So there are a bunch of things it's doing. We don't know all the details yet, but we have some idea. These processes are all things that can work in a human or a mammal, potentially. And so the next step, of course, does it work in mammals? We looked at the mouse. We ran one of these 3-year studies. And it works. Both the hao knockout in gray extends that curve to the right, and putting 3-HAA in their diet extends it. So this is where we're at with this. Is our probably leading candidate in my lab that we're working on. And we have found an effect where we started in the worm, we screened some genes, we found a gene and a molecule we think is going to benefit aging. It works in the mouse, and now we do need to do more work to figure out, okay, is it doing the same thing in the mouse, how robust, how safe it is. But ultimately, if this continues to look promising, this could be a candidate for moving into human clinical trials. So does it work in humans? We don't know yet. We've gone through, let's take a step back to our pyramid. We've gone through the discovery process. We've identified what it's doing. That's what we call the mechanism. Preclinical validation, that is the mouse work. So we know it works there. We do have more work to do here, and we don't know if it works in humans. That is the final step. It turns out we don't know for sure that anything works in humans. I told you the field is young, and I mentioned the first clinical trial. Let's look at that. This was just a couple years ago. This clinical trial is, I think, still just in the early stages of recruitment, might even be pre-recruitment. But we do have our first ever test. This is really exciting. So up until a few years ago, we could test a gene for heart disease. We could test a gene for drug for cancer. We could test a drug for Alzheimer's disease. But there wasn't even an indication at the FDA for aging. And now we have that for the first time. And what they do is they take advantage of this idea that aging is driving all of these diseases. And so they start with 5 diseases that are kind of unrelated in their molecular features. So dementia, heart disease, diabetes, cancer, and I guess Alzheimer's. I guess there's another form of dementia in there. And what they do is they start with people at risk for multiple diseases. We're going to give them this drug, metformin, for 5 years, and we're going to see how many of those diseases they got. So this gives us a lever where we can ask, if the people get less on average diseases, that suggests we're affecting the aging process. Metformin is a really great drug. How many of you know someone, or you yourselves maybe are taking metformin? There's a lot of people, right? This is part of the reason this drug was taken. We've been using it for 50 years. It's a diabetes drug. It's very safe. So especially for the first drug, you want to make sure there's no chance of harm. We know how it works in humans. But there's also this interesting feature that if you look at patients with diabetes taking metformin, at least in some ways, they look healthier than non-diabetic patients who are not taking metformin. So that suggests that maybe it's doing something beyond fixing the diabetes. There are other drugs that are getting close to this point that are looking promising. Okay, so we're getting toward the end. We've talked about why basic science is important. I want to talk about just briefly what you can do today to age better. So we say there's no drugs available, but does that mean there's nothing we can do? And that's certainly not true. So how do you age optimally in 2026? This is going to be a little mundane, I'm sorry, but you can sleep better, you can eat better, you can exercise, and you can spend time with each other. Community is super important. The reason I mention these is because there have been lots of studies of these things that we kind of, your doctor has been telling you for years, right? But we know that all of these things affect many of these molecular signatures of aging. And what I would argue is, if we do end up having a drug that can extend lifespan and push back these diseases, it'll work better if you have your basics in line first, and you have kind of these things as optimal as you can get. So we can do that today. There's a more medical-oriented things. I am not a medical doctor. I am recommending you talk to a medical doctor, just to be clear. But take supplements, but don't take every supplement you see on YouTube. Take supplements to correct deficiencies. So you can get tests for vitamin D and vitamin B and all of these kind of standard things. Most of us are deficient for vitamin D. We know that at a population level. I grew up in Seattle. There we don't really see the sun. In Tucson, we hide from the sun inside, and then we put on sunscreen, so that also stops you from getting the vitamin D. So it's even true here where we get a lot of sun. Keep up with your screening. We have screens for colon cancer, for breast cancer, for various other diseases. These are there for a reason. The whole idea behind aging science is if you catch something earlier, it's easier to treat. That's true even today. So if you can catch these diseases earlier, you're much more likely to have a good outcome. And then, of course, stay curious. This has a kind of a dual meaning. I keep informed by the field, but also just continuing to learn is great for brain health and brain aging. So, dual meaning. Where can I learn more? So if you do want to learn more, there's lots of resources. Right here at the University of Arizona, I have a couple of recommendations. Within the College of Science, we have the Biosphere 2. Now, these days, if you haven't been there, they do more environmental research, which is interesting. But they have this history with aging research where it was originally built as a contained habitat where people could go in and live, and they were trying to sustain a human colony inside this thing. And it turned out they accidentally dietary restricted themselves. So this was the first human trial of dietary restriction, inadvertent or otherwise. So there's kind of a fun history with aging there. They're fine. They actually came out probably healthier than they went in for it, but maybe less happy. My lab is in the Bio 5 Institute up on North Campus. Every year they do a Discover Bio 5. One of the pillars of Bio 5 is aging science. And my lab is very active in tours. You can come look at the worms, see the robot, that sort of thing. We have not one but two centers that are focused on aging. One is the Arizona Center on Aging. This is more, it's biology, but it's also clinical medicine, nursing, that sort of thing. And then the College of Pharmacy also has the recently named Coit Center for Longevity and Neurotherapeutics, looking for drugs that can affect Alzheimer's disease and brain aging and longevity. A couple of podcasts. I mentioned Matt Kaberlein was the one with the map analogy. He runs a podcast, very science-focused. There's a lot of hype and a lot of bad information out there in aging, in the public-facing aging science, for better or for worse. People are excited about this, but it's also hard to find necessarily good information. So these are things I have personally listened to, and they're science-backed, so this is a good place for really understanding what's scientifically legitimate. The Buck Institute's a private institute on aging. They also have a podcast, We're Not Getting Any Younger. And there's a bunch of books out there, Why We Die, Ageless. Methuselah's Zoo is a fun one by Steve Ostad. It talks about how we use very long-lived species like turtles and whales and clams to learn about ways that we might live longer. You can participate in citizen science. There are different ways you can do this. The NIH has an All of Us program, which I'm personally signed up for. They're trying to take 1 million plus Americans and just see how they change, how disease develops with age. It's not just aging focused, but that's part of it. And really just understand at a very large population level the biology. And you get a bunch of information about your own health and medicine at the same time, if you're comfortable with putting your data into a research fund. The other side is the Dog Aging Project. This has a couple of goals. Just like the All of Us program, the one goal is to understand how dogs age. So why would we look at aging in dogs? I want my dogs to live longer. That is actually a big part of it, right? So that it's motivating for people to get involved. But they also are close to our biology, and they share our environment. So they are, in many ways, a really good system for understanding an aging model that is not as long-lived as us. They do not live as long as we do, so we can learn things faster. But they also share a lot of the same factors that influence aging. There's also a small clinical trial of about 600 dogs testing a different drug. This is a clinical trial. Veterinary clinics are considered full clinical trials and require similar regulation for a drug called rapamycin, which is another one of the promising first-line drugs for aging. There are many institutions as part of this. University of Arizona is one of them, and we have a specialty center on canine cognitive and behavioral aging. I have two dogs. This is Doppler and Zelda again. They've both been through the program. So I know, we can just leave that up there for a minute. So. If Zelda were here, she'd be trying to sit in every one of your laps right now. The other thing I would say is to just share your enthusiasm. You're here tonight spending your own time to learn about current science. Share that enthusiasm with your family, with your friends. Regardless of your political affiliation, talk to your representatives. Basic science, because it's so far from a product, companies are a big part of doing science and bringing discoveries into product, but they tend to come in later when we know more about a system, and then they're willing to invest because they can develop a product and sell. Earlier on, discovery research, basic science, that is more of a public effort. It's taxpayer supported. And so talking to your congressman and your senator, there is benefit in that. They do listen to their constituents. If you support science, communicate it to them. That is an important part of advocacy if you would like to so do that. So I hope tonight that I have given you some enthusiasm about the aging science. I'm very optimistic about the field. We're on the cusp of knowing whether some of our early discoveries are going to work in humans. And while we have a lot to learn, we know a lot, and there's a lot of things building their way through the pipeline. I think this field is going to change more than any other. I mentioned the companies. There are new companies popping up every day. There's a huge investment from the private industry into this field. And then driven in part by the basic science that we're doing at the University of Arizona and other universities in the academic space. With that, once again, thank you for supporting science and being very much. Good evening.
I'm going to let the audience know that you're going to...
Sounds great, thank you.
So let's just thank George for what a fun, engaging, and informative talk. And George is going to hang out up here at the end if you want to come up and ask him questions. I have a couple of things to announce and share as we close up our evening. First of all, I just want to invite you to next Wednesday's lecture series, which is featuring Dr. Martha Bhattacharya from the Department of Neuroscience. And Martha is going to share a little bit more about her talk for next week in this video.
Hi, my name is Martha Bhattacharya, and I hope you will join us next week at Centennial Hall for the second lecture in the 2026 lecture series. In my talk, we will discuss the genetic relationships between insects and humans and how we can use these similarities to understand how evolution has built brains that can think, feel, and make decisions. I'll share how our research has made discoveries using insects that reveal clues about human brain and nerve disorders, including dermatitis, epilepsy, and more. I'd love to see you there.
Well, thank you for sharing your love for science with us and spending this evening on our 20th anniversary of the lecture series. And for all of you who are basketball fans, I just want to give you another little teaser about next week. I'm sure you're excited about this year's basketball season. Who isn't right now? But we're going to have a special guest here with us next week from Arizona's men's basketball, and so you won't want to miss that. And we're looking forward to seeing you then. So please get home safely and see you next week.